Good morning to each and every one of us. My name is Roger Optional, and today's video I'm going to discuss about genetic and man. So in this topic, we will be able to know what are the function of genetics in terms of human diversity, or able to discuss humanity into genetics based on reality. So the subtopic of this lesson is all about to identify or to differentiate what is cytogenetics. Next is to identify the sum of inborn errors of metabolism. And last is to differentiate also the behavioral genetics. So the lesson objectives is to differentiate what is cytogenetics, and we will be explore more about what is cytogenetic based on in our reality, and to identify some of the inborn errors of metabolism in terms of cytogenetics, both in genetics and human behavior, and to determine what is behavioral genetics through in our humanity, and how we going to deal, or what are the functions to provide, what they're going to do to explore more about this topic. So before we come up or discuss what is cytogenetics, we will be able to know who is the man behind, or the people behind, or the take of people, or the person who identify or being a cytogenetic discussed more in reality. So first, we have to identify who is the father of cytogenetics. So first, Walter Fleming from 21st April to 1843 to the 4th August 1905 was a German biologist and a founder of cytogenetics. In addition to that, he was born in Schanzenberg, now part of Schwerin, as the fifth child of the only son of the psychiatrist Carl Friedrich Fleming from 1799 to 1880, and his second wife, August Winter. So next, in a broadly sense, what is cytogenetics? So cytogenetics is the branch of genetics that studies the structure of a DNA within the cell nucleus. So it based that the cytogenetics, it is the study of the structure of the DNA. So we have to sense that the structure of the DNA are combined into the cells of the nucleus. In addition to that, the DNA is condensed during cell division and form of chromosomes. So it means that during the DNA, in terms of genetics, we are condensed during the cell division and form into chromosomes. So in line to that, we will be able to discuss the chromosomes in line to cytogenetics, or how we be able to deal the chromosomes and DNA in part of the cytogenetics. So the cytogenetics studies the number and morphologies of chromosomes. The cytogenetics studies the number and morphologies. So which means that we will be able to deal the numbers and the forms of things that in a particular living organisms. So using chromosomes, binding techniques, classical cytogenetics. So using the forms of chromosomes, we will be able to form a classical cytogenetic to be able to discuss more about what is the chromosomes in terms of cytogenetics. So are the hybridations fluorescently labeled probes or molecular cytogenetics. So the number of morphologies of chromosomes in a cell of a particular species are always constants. So in a specific cytogenetic part, there is a specific species that can be able to in line to being constant in the particular species in the chromosomes. So in addition to that, in the most cells of the body, with the exception of reproductive cells and others such as the liver. So this characteristic of this each species in humans, such as the number of chromosomes, is 46. So we all know that the human, both male and female gender, we have X and XX and XY chromosomes. We have total the male, we have XX 23 chromosomes, and the both male, we have XY 23 chromosomes. So next, in a basically or the simplest way of meaning of cytogenetics, Cytogenetics is the study of chromosome and its related disease states caused by abnormal chromosomes number or structure. So we will be able to study about the chromosomes and related to diseases stated caused by the abnormal chromosomes. Say for an instance, there are things that there are many people, so marami tayong tao na yung nag-a-adapt ng yung maling yung chromosomes na hindi umaakibat sa ating sarili. So that's why nagkakaroon siya ng diseases. So parang maling pagfa-function na siya ang narasa ng isang tao. And the chromosomes, number of chromosomes, the cytogenetics involves the study of chromosomes. So, yun nga. So, ang cytogenetics is nag-rely siya sa pag-aaral ng chromosomes. Hindi lang sa pagbabase ng mga genes or the multiple allele na ma-adapt natin sa ating sarili, but it's in rely to sa chromosomes talaga, yung mga particles na nabibase natin sa ating sarili. So, chromosomes in cytogenetic terms is a complex structures located in the cell nucleus composed of DNA. So, we already know that the chromosomes is a complex structures that composed of the cell nucleus. So, we will be know about this composed of DNA, the histons and non-histons proteins, or the RNA and the polysaccharides. So, the things that the cytogenetics will be able to know that the chromosomes are in part of this lesson. So both the number of the structure of the chromosome are analyzed. So the things that we will be able to analyze the structures of chromosome in terms of cytogenetic. So next, so the chromosomes in cytogenetics. So first, we have each chromosome is composed of two sister chromatids. Next, each chromatid represents one molecule of DNA. Then next, each chromosome has a unique J binding pattern that is constant from individual to individual. So for the illustrations, this is the chromosome. We have two sisters with chromatids. So as we can see in the picture, these are the centromere and this is the both homologous chromosomes. So if they combine both of these, we have, if there are two, so we can make a sister chromatids at the same time. We have also the left side, the sister chromatids also. So this is the illustrations of the two sisters with chromatids in chromosomes. Next is the chromosomes with one molecule of DNA. As we can see in our illustrations, so in this part, we have centromere, and this is both chromatid. So this first chromatid, yellow and blue, symbolize the molecule of the DNA, and the left side is also an DNA. So they are composed of both single molecule of DNA. Next is having a G-band pattern. So as we can see, this illustration is chromosome having 4 G-band pattern. So first we have centromere, and the human, and the chimpanzee, and the centromere also. So this is the chromosome having G-banding pattern. So the conventional cytogenetics. So the conventional cytogenetics cells are stimulated to divide are incubated for 48 to 96 hours. So in this cytogenetics, cells are stimulated, which means we can be able to divide it or separated or incubated during 48 to 96 hours. So it's that, I think it's almost 2 days. So cells are arrested at mitosis. So we all know that mitosis is a type of cell division that results in two daughter cells of each having the same number and kind of chromosome. So dito parang nagre-rely siya yung cell sa isang cytogenetics is nagre-rely siya sa kanyang true mitosis rather. So next, conventional cytogenetic cells are dropped into glass slide and G-banded. So parang ganito siya. So sa kanito na yung structure, makikita natin na parang siyang cylinder na parang dito siya na umaaga siya, pababa dito siya. So I think ito yung the drop. So parang magulo siya. Something like yan. So, into the glass slides and G-shaped band. So, what tissues are appropriate for chromosome study? So, a tissue that can be simulated to undergo cell division in vitro. So, that means that vitro means that perform organism cells or biological molecule outside their normal biological context. Next, it is only during mitosis of the cell cycle that inside chromosomes can be visualized with a light microscope. So, dito nga, during mitosis, makikita natin yung cell cycle. So, distinct chromosomes that can be visualized through light microscope. So, parang unseen siya, dapat ginagamitan siya ng particular na bagay para makita light microscope. So, we all knowing that na ang microscope is makikita ng mga kahit anong maliliit na bagay na mabibigyan talaga ng buhay kapag nakikita mo dyan, kapag ginagamit ang microscope, kapag we have to identify na organisms. Next, example of tissues that can be studied is chorionic villi, then ammonic fluid and peripheral blood and skin and bone marrow. So those are the examples of tissue that can be studied in appropriate tissues used in the chromosome study cytogenetics. So there are things also in identify the chromosomes in terms of cytogenetic. So first, to identify chromosomes, we have to identify first their size. So their size, kung ano ba sila, yung maliit ba sila, or malalaki ba sila, or tiny or little organisms na mababasa natin in line to cytogenetics. So they're also based with their shape, so the position of the centromere. So yung parang makikita natin kung ano ba siya, or below ba siya, or a triangle ba siya, or square that can be studied. Then there are also the NB chromosomes are flexible. So which means yung NB is neuroblastoma, which means are flexible. Para siya yung that can be easily yung kapag ang DNA is kapag pinaseparate siya, ganyan isineseparate siya, para siyang elastic, so para siyang yung guma na ginagamit. So parang ganun siya, is flexible kasi siya. So I think na parang malambot siya kapag something is in reality siya. So also, the bonding patterns produced by specific strains or the desma, yung pattern na G bonding patterns, yun isa din yun sa to be able to know sa pag-identify ng chromosomes in terms sa cytogenetics. Then, in line to that, chromosomes are analyzed by organizing them into karyotype. So, karyotype there describes that there are the things na parang an individual collection of chromosomes used to look for abnormal numbers or structures of chromosomes. Parang nagbabase siya kung ano yung na-organize ng isang chromosome sa isang specific na cytogenetic part. So next is the cytogenetic methods. So the cytogenetic methods involves of examinations of chromosomes. So in this part, we will be able to know kung ano yung function or ano yung essence or value talaga ng chromosomes in line to cytogenetics. And it is also to identify structural abnormalities. So, parang identify. So, parang yung nag-study siya, or parang inaalam niya kung ano yung mga abnormality structures na nagpa-function ng chromosomes sa cytogenetic time. So, chromosomes also of dividing human cells can be analyzed clearly in a white blood cell. So, parang ina-identify ng isang chromosomes through blood cells, specifically the lymphocytes, which are easily collected from the blood. So next is the purpose of cytogenetics. So the cytogenetics involve testing samples of tissue, then the blood or bone marrow in a laboratory to look for changes in chromosomes. So dito masasabi natin na ang cytogenetic talaga ay isang pag-aaral kung saan malalaman natin ang mga sample tissue ng isang tao or isang organisms para ma-identify natin siya kung meron ba siyang mga abnormalities na nakikita sa kanyang sa kanyang sarili or an organism. Kasi kapag mayroon tayong makikita ng mga abnormalities sa organism sa isang organism, matatawag natin that can be tulig na magkakaroon siya ng sakit gaya ng isang tao. That can be yung may maraming mga abnormalities. So I think na isa yun sa hindi na na testing samples ng isang laboratory para matingnan yung changes ng isang organism sa kanilang chromosome. So, including broken, missing, or rearranged of our extra chromosomes. So, changes in certain chromosomes may be sign of a genetic disease or condition or some types of cancer. So, yun nga. So, nagkakaroon siya or naglimitahan so pagkakaroon ng isang sakit sa isang tao or an organism.